Texas Lutheran University.
So it's our pleasure to have here today Dr. Jerry Brudvig from Yale. Anybody know where Yale is? I'm preempting some of his slides, but I want to. It's in Canada. No. South of San Antonio, exactly. And he's going to talk to us a little bit about. He said about 50% biology, so I'll kind of snap through that part, and then 50% chemistry, a little bit about water oxidation by Photosystem 2 and artificial systems.
Thank you.
Thank you.
Thanks, Phil. Thank you. Thanks for the invitation. It's great to be here. And I'm going to talk, as Bill said, about Photosystem 2, which is the biological system that uses light to oxidize water. It's important for energy conversion processes, which we're interested in. And we're trying to emulate that by developing artificial processes. So I'll focus on the artificial, more inorganic oriented in the second half. So, Bill, you can sleep the first half and wake up the second half, and if you're interested in biology, you can watch the first half and sleep the second half. But, so Bill asked, where is Yale University? How many, have you probably maybe heard of Yale? But have everybody heard of Yale?
New Haven, Connecticut.
New Haven, yeah. Where's New Haven? Does this work here?
Turn it on.
What do I have to do?
It popped out of.
Oh. Okay, it's not in presentation mode.
Yeah.
There we go. Okay, now it works. So, where is Yale on the map here? Way up in the upper right? There it is, right there. It's in New Haven, Connecticut. We're about an hour from New York City. Yale's one of the Ivy League schools. It's one of the oldest schools in the United States. It was established in 1702, so we're more than 300 years old. If you go there, if you ever visit, you'll see lots of Gothic, buildings look like they're 300 years old, but we do have some nice facilities inside those old-looking buildings. So it's a great place for grad school. If any of you are interested in chemistry grad school, we have a very, very good.
Dr. McCluskey go there?
He did. Yeah, we talked about that one.
Yeah. He worked for Ken Weiberg, I think. Yeah. Ken is still around, but he's been retired for a number of years now. And so I want to talk about energy, and particularly related to photosynthetic water oxidation and artificial water oxidation. Some of you may know Rick Smalley, from Rice University. He was a Nobel laureate. Buckyballs. He discovered this new form of carbon. And towards the end of his life, he's no longer living, but towards the end of his life, he was a champion to raise awareness of the importance of energy. And this is his list of humanity's top 10 problems for the next 50 years. He put energy at the top of the list. And one reason for that is we're going to need more energy in the future. The population is growing from 6.5 billion in 2004 to probably around 10 billion in 2050. But energy is also very interrelated. So if we had enough energy, we could desalinate the oceans and have unlimited amount of water. And if we had an unlimited amount of water, we could irrigate all the fields and grow as much food as we need, and environmental impacts and so on. So energy percolates and it's important all the way down the list. And so this is a big topic of our interest. And as you know, most of the energy we use now comes from burning fossil fuels. And that's a big problem for the future. We need to come up with a carbon neutral way of powering our planet. And I think by most people's assessment, solar energy has the highest capacity for providing power. And so our research is directed towards trying to understand the molecular processes whereby we can convert solar energy into storable form. So that's a big problem, a big requirement for solar energy, because right now most of the solar energy that's used is used in solar panels, photovoltaic panels that produce electricity. But electricity just feeds into the grid and it's instantaneous. So the amount of electricity we produce has to always constantly match the demand. So if the demand goes up, we have to produce more electricity. If it goes down, we have to produce less. And so you have to have the capacity to ramp up and down quickly the production of energy. And that doesn't work for solar. Solar, the sun is out and it gives you what you get, and it's not there at night. And the same is true for wind. You know, wind is blowing or it's not, and you can't just turn it up if you need to. And so how can you get around that? You have to be able to store. And so there are different ways we could store electricity or solar energy, and one is using batteries, and that's a good thing, but it's also very expensive and they're very heavy, and so we take an inspiration from nature. Nature stores solar energy in the form of chemical bonds by making fuel. So this is a field that is growing right now called the solar fuels field, which is an effort to try to take inspiration from nature on how to use sunlight to make fuel, to store energy in chemical bonds, but to do it in artificial processes that hopefully could be more efficient and more long-lasting than photosynthesis. So I want to start by talking about natural photosynthesis, how plants do solar energy conversion, particularly water oxidation, and then we'll shift gears halfway through to talk about artificial processes. So this is a picture of a cornfield actually just down the street from where I live in Orange, Connecticut, which is a town just outside of New Haven, Connecticut, where Yale is located. And here's a state park down the road a ways. But what plants do in photosynthesis is they take these low energy molecules, very stable molecules, carbon dioxide and water, and use light to provide the energy to break them apart and turn them into high energy molecules like sugar. This is the chemical formula for glucose, which is the initial product in photosynthesis. But ultimately, all biomolecules, all cellulose, everything else that plants make come initially from the fuel of sugar molecules. And then oxygen is produced as a byproduct. Actually, that's a waste product from the point of view of plant photosynthesis. But it stems from the fact that, well, maybe let me go back. It stems from the fact that this is an oxidation reduction reaction. So water is oxidized and carbon dioxide is reduced to make sugar. And so that's the fuel is the reduced product, oxygen is the oxidized product. Now, this reaction has been going on for billions of years. The Earth initially didn't have any oxygen in the atmosphere. About 2.5 billion years ago, the atmosphere started to have oxygen introduced into it. And it came from this reaction. Now we have about 21% of our atmosphere is oxygen. And that essentially all of it came from photosynthesis. And it's not changed much over the last 100 million years. We're pretty much in a steady state where the amount of oxygen produced in photosynthesis is pretty much balanced by the amount that we use and other organisms use in respiration. So in the summertime, plants are making oxygen. The oxygen concentration is going up a little bit. Carbon dioxide is going down. And in the wintertime, it goes the other way, and it's pretty much balanced from the natural processes. But over the Earth's history, of course, there's been an excess amount of oxygen production, so we have an oxygen-containing atmosphere. And I did a calculation. The amount of oxygen in the atmosphere required an amount of water to be produced on the reactant side that would lower the ocean level by 4.8 meters. It's a heck of a lot of water that got oxidized to produce this amount of oxygen. And so how many of you are taking general chemistry now? Anybody? Or how many have already taken general chemistry? What's the first thing you do in general chemistry? You balance equations, right? Stoichiometry. So this is stoichiometry on a global scale. So if we've got 21% of our atmosphere is oxygen, that has to be balanced. The stoichiometry has to be balanced. A lot of carbon dioxide and water got used up, this much water to lower the ocean levels. But what's the rest of the equation? That's biomass. And where's that in the biosphere right now? That's the fossil fuels that we've been digging out of the ground. Now, some of you might have heard people predicting that we're going to run out of oil. Well, I'll tell you right now, we will never run out of oil, and absolutely never. And why is that? Well, we've been burning fossil fuels for hundreds of years, and how much have we changed the atmospheric oxygen concentration? Essentially zero. Barely detectable, 20 parts per million out of 21%, it's a negligible amount. What's that telling us? This is just stoichiometry. It's telling us that the fossil fuels buried in the ground, we've barely scratched the surface of them. And there's going to be essentially inexhaustible supply. It's not infinite, but the limiting factor is not going to be running out of oil. It's going to be, we're going to destroy our environment if we actually burn that much fossil fuel because of the carbon dioxide we produce. we're never going to lower the oxygen concentration in the atmosphere enough for us to actually have a hard time breathing. You know, we couldn't possibly, environment would be destroyed. So we need to come up with a way to power a planet that doesn't involve burning these fossil fuels. That's one of the motivations for my own research. And one of the goals in that research is to try to learn how plants do this, how these corn plants and trees actually do the process of using light to make fuel. And so that's what we're looking at. And so my talk today will be two parts. Photosystem 2, which is a membrane protein complex that does light-driven water oxidation. I'll try to tell you a little bit about that. I'm not going to go into all the technical details, but to give you kind of a broad overview of this system. And we've been working on it for many years. And then I'll shift gears to talk about our efforts, which are more recent. Only in the last 10 years or so, we've been starting to work on developing artificial processes, particularly focused on the synthesis of artificial catalysts, so inorganic compounds that can catalyze water oxidation, but then applying them to do photochemistry for making fuels. And I want to also emphasize that this is a very collaborative project. We work very closely together with two of my colleagues, Victor Batista, who's a computational chemist, and I'll talk a little bit about the synergy between our experimental work that my group does and the computational modeling that the Batista group does. And then that's more in the first half when we talk about Photosystem 2. And then also Bob Crabtree, who's an inorganic chemist, and we work closely, have many years. I sometimes tell people that I'm not an inorganic chemist, although my department now categorizes me as an inorganic chemist. I teach inorganic chemistry now, even though I've never taken a course in inorganic chemistry. I learned everything I know from Bob Crabtree. And if you take a more advanced inorganic course, you might learn about organometallic chemistry. The book was written by Bob Crabtree, so if you take organometallic, you'll probably use his book. So that's what I'll cover. So let's start out with Photosystem 2. So for the biologists of you in the room here, here's a leaf. If you look at the single cells, you'll find a chloroplast. And if you look at an electron micrograph of a chloroplast, this is what it will look like. And you see inside these structures, they're called thylakoid membranes. These are membrane structures that contain proteins, and those proteins bind all of the pigments, the chlorophylls and carotenoids that give the color to plants' leaves. And these are all embedded within a membrane, depicted in a cartoon fashion here. And they function to do the photochemistry in photosynthesis. So they absorb light, and they use light to initiate electron transfer reactions. The initial step is called a charge separation, as an electron is moved from one site to the next. And the first step in that occurs in Photosystem 2, and it's abbreviated PS2, and that stands for Photosystem 2, not PlayStation 2. And what it does is it takes light to produce a charge separation, and then uses that charge separation to oxidize water. So that's the initial event where electrons are extracted from water. They're passed down a series of reactions through the so-called photosynthetic electron transport chain, ultimately produce this very strong reducing agent, NADPH, which is an organic reductant that is providing the reductant that's used to reduce CO2. So this is, you can't hardly see it, but this is the carbon dioxide fixation cycle up here, which is sometimes called the dark reactions because those don't need light. But they do need the high energy, strong reductants that are produced by the light reactions. And that ultimately really produces sugars, and they're being exported out here. So that's the overall process of how photosynthesis works in a very quick spin through. You probably see this in more detail if you take biochemistry, and maybe that's what you teach. And we're going to focus on this Photosystem 2 complex, which is a fairly large membrane protein complex, and this is a picture of the crystal structure of it. So this is what this is really an enzyme. It carries out an enzymatic reaction to take water molecules to convert them into oxygen, and of course that generates hydrogen ions and electrons, and those are used to reduce an organic molecule. PQ is plastoquinone, and this organic molecule is converted to its reduced form, or plastoquinol, and that ultimately then diffuses along and carries electrons along the pathway. So that's the overall process, but we're going to focus on this manganese catalyst. This is the catalyst that does the water oxidation reaction, and it's buried deeply within this protein complex. So you sort of see a little glimpse of it here. It's called the OEC, or oxygen evolving complex, And it's in my opinion, one of the most important catalytic sites in all of biology because it catalyzes the oxidation of water. And this is an incredibly difficult reaction to carry out in artificial systems. We've been really challenging to develop artificial systems can do this. Nature does it beautifully well, so we'd like to understand, how does that work? Now, a big breakthrough in this was the crystal structure of Photosystem 2. This was reported, published in Science in 2004. That's quite remarkable because this is a very large complex of 20 protein subunits, a lot of chlorophylls, these little green things inside, and carotenoids, these orange things. There's almost 100 cofactors in Photosystem 2, so there's a very complicated large protein complex. These rods represent transmembrane alpha helical structures of the protein as it snakes through the membrane. And it provides a scaffold to hold all these pigments that absorb light, and they funnel that excitation energy to the center part, which we call the reaction center, and that's where a charge separation occurs. So I'll show that on the next slide. So that's the overall process. And this was a remarkable achievement in 2004, this crystal structure of Photosystem 2. But it has one problem, and I'll come to describe that in a little bit more detail. But X-rays are ionizing radiation. So high energy radiation. If X-rays strike a molecule, they break it apart. And that happens by ejecting an electron out of molecules, like a water molecule. And so X-ray exposure to any type of material causes electrons to be ejected and flying around in the sample. And that's a big problem for high valent manganese centers, is that those manganese ions get reduced very quickly under X-ray exposure. This is a radiation damage problem. And what that resulted in is that although you could see the structure of the protein part pretty well, the structure of this oxygen-involving complex was not very clear. So I'll come back to that in a minute. So let's look at the initial events now. This is something that happens on a very, very short time scale. So light is absorbed. It is by all these chlorophylls that are present in the Photosystem 2 complex. And that excited state gets transferred to this cluster of chlorophylls. And this is a very special cluster of chlorophylls because in its excited state, it doesn't pass the excitation energy to another molecule, but it initiates a charge separation reaction. So this electron transfer. This is the initial event where the energy of a photon of light is converted to chemical energy, so the energy of an oxidized and reduced pair. Of course, you've now generated a very strong oxidizing center, a chlorophyll that's lost an electron, so it's a chlorophyll cation radical, and a reduced molecule. In this case, it's a pheophytin, is an organic cofactor, which is very strongly reducing. So you put a really strong oxidant next to a really strong reductant, and what's going to happen? They're just going to recombine. And that's the process that you want to avoid if you want to use solar energy efficiently, is to minimize that or prevent it. And the way that happens in photosynthesis is to move the electron away as fast as possible and move the whole or oxidizing equivalent away as fast as possible to create now a very large separation between the two so that they don't recombine and have a very long-lived charge separation. And that's what these photosystems do in plants, do incredibly well. They do this with a quantum efficiency of virtually 100%. Every time a photon of light is absorbed, a stable charge separation is produced. And that's something remarkable. We have a really hard time reproducing this kind of efficiency in artificial systems. The end result is that we've oxidized this manganese cluster, which is then ultimately going to oxidize water. But you remember stoichiometry. So this process of charge separation is a one-electron reaction, one electron for one photon. But you know that in order to oxidize water, you need to do a four-electron reaction to produce oxygen. So this process has to happen 4 times in succession, 4 photochemical charge separations in sequence. And this is really beautifully illustrated if you measure the yield of oxygen of a sample. This is from spinach membranes. And so giving single turnover flashes. So measure the yield, and after the first flash, there's no production of oxygen. Second flash, no production. Third flash, there's a big burst, and then every fourth flash afterwards. So this period 4 cycle, which is telling you the stoichiometry of photons to oxygen. You need 4 photons to make 1 oxygen. And this is represented in terms of a sequence of intermediate oxidation states with a subscript to denote how many steps have advanced. So these are called store states. Stored 0, 1, 2, 3, 4 equivalents, and now you have enough to oxidize water, and you can make oxygen. And this is the so-called Koch cycle for oxygen production in photosynthesis. Now, maybe this could provide a sort of inspiration or maybe a challenge to this, but this paper that was published by Bessel Koch, who's a Dutch scientist, was the first author on that was an undergraduate researcher named Dick Forbush. He's actually a professor at Yale now, so I know him. He went to the dark side. He's now a professor of medicine, so he's in the medical school. He went from plant photosynthesis to medicine, but he still does very good research. But this paper is maybe his most famous paper, and he did it as an undergraduate researcher. So you can do some pretty good things doing undergraduate research. So what my research over the years has been aimed at is trying to understand at the molecular level what's going on. What are the chemical transformations as we go around this cycle? These represent oxidation states of this tetramanganese cluster, and they're very high oxidation states. So the dark state, the so-called S1 state, which is the state that's stable in the dark, contains 2 manganese 3 ions and 2 manganese 4 ions. So if you've taken some inorganic chemistry, maybe in general chemistry, you may know that the stable oxidation state of manganese in aqueous solution is manganese 2. So this is already 6 electrons oxidized above the ambient oxidation level of manganese. That's really highly oxidized just to start. That's why it's so susceptible to radiation damage when you shine X-rays on, because it's very highly oxidized. But that's not enough yet to oxidize water. In fact, this is the dark stable state. You have to go 3 steps higher before you've reached a high enough level of oxidation to actually make a water molecule react with another. That's really something that's hard to do. And so that's what we'd like to understand. Now, I'm not going to go through this cycle in any detail, but what I'll do today is just talk a little bit about the structure in the dark state. And we've done work to try to use the crystallographic data that were published to try to refine it and connect it to experimental spectroscopic and other data that we've obtained to try to refine and develop the best model we have of the structure. I'll describe that. And then I'll jump ahead to what's going on in this last step. How do we think this reaction is happening to make oxygen out of water? Okay, so let's go back to 2004. I told you this landmark result that was published in Science with the crystal structure of this very large membrane protein complex. And if you look at the electron density of this manganese cluster, you just see a big blob. So if you have a crystal structure, you should see each individual atom, but you can't here because the system suffers from the radiation damage problem. So we can't resolve the individual atoms. So we can't really see the structure. And of course, this is the part that everybody's most interested in is the catalyst, and you can't see it. So at that time, I approached my colleague Victor Batista, and he was a young assistant professor, and I said, could we use computational methods to refine this structure? So we've got a pretty good model of the protein part, and could we refine the structure of this part we can't see by crystallography using computational methods? That's a really challenging problem, and maybe wasn't quite, didn't quite appreciate how complicated it was, but he said, oh, that sounds great, let's do that. So we started working on this, and have been now up until the present time. This has been the last 12 years. And Victor's now one of the most expert people on this problem, although he knew nothing about it when I first started talking to him. But we started working on it, and 2 years later, we published a paper. And so what we were doing was we're taking quantum mechanics, using a quantum mechanical model, using density functional theory to calculate the structure. This is something that a very large calculation, and we calculate all the manganese plus the protein groups that are coordinating it as a quantum calculation. And then we modeled the rest of the protein using molecular mechanics. So molecular mechanics is just using Newton's law of motion of how things move around. So that's relatively unsophisticated or uncomplicated, but it takes into account the rest of the protein. And so we use this method, quite well known. It was recognized by the Nobel Prize a few years ago, the development of this QMMM method. And we were the first to apply it to this large complex of photosystem 2, and we published a paper proposing this cuboidal structure where there are 3 manganese and a calcium in a cube bridged by oxygens, this manganese oxide cube, and then one attached manganese, this so-called dangler manganese, that had water molecules coordinated to it, and also the calcium having water molecules coordinated to it. So this was a proposal we made from computational modeling. I, at the time, was pretty convinced that the crystallographers would never resolve all the atoms in a crystal structure, but that wasn't the case. In 2011, a group from Japan, John Ren-Shen's group from Okayama University, published a very high resolution at 1.9 angstrom. It was an extremely good resolution for a large protein structure. And it resolved every atom in the oxy-evolving complex for the first time. Now, this was recognized as one of the top 10 breakthroughs of the year by Science magazine. So the Science magazine, the first issue of the year, always has a breakthrough of the year and then the top 10 runners-ups. And this is quite remarkable considering that Photosystem 2, the crystal structure of Photosystem 2 was published more than a decade before. And nonetheless, it was a breakthrough of the year. So why was it? It was because it's the first time this manganese cluster was fully resolved. So everybody thought, okay, problem solved. And people who see crystal structures, if you read the literature and you see crystal, okay, that's the holy grail. Now we know it. Well, wait a minute. And so we started looking at the crystal structure, and this is now taking this cluster of manganese ions and looking at the bond lengths between the manganese and oxygens. And we've done a lot of model chemistry with manganese and oxides. And we know that a high-valent manganese oxide bond length should be 1.8 angstroms. And some of them are. This one is 1.8, this one's 1.8. But many of them are really long, 2.4, 2.5. Those are way too long for the bond lengths you should get for manganese oxide, high-valent manganese oxide clusters. So what does that mean? Well, the biochemists, not really knowing any inorganic chemistry, said, well, that's because this is a really high-energy oxide, and it's going to be ready to react to make oxygen. And that doesn't make any sense. Bond lengths, you cannot make bond lengths different than what they are. Chemicals are chemicals. And so to us, it looked like this was, in fact, not the high oxidation state that it was supposed to be in, but it been, in fact, reduced. And we concluded that they hadn't avoided this radiation damage problem. They had just avoided the disorder that happened after the metals were reduced. And to test that, we then did some computational modeling. So John Ren-Shen first reported this at a congress in Beijing in 2010. And right after his talk, I went up to him and said, would you want to collaborate with us and do computational modeling of the structure? And so he gave us the coordinates about a year before they were released publicly, and we started working with them. And the Batista group then took those coordinates and asked, is the structure consistent with spectroscopic data? And so this is a technique that we compare called extended X-ray absorption fine structure spectroscopy, or XAFS. And the question was, do the coordinates match the XAFS data? Now, I won't have time to go into what XAFS is, but it's a technique that doesn't produce radiation damage because it can be done with very low exposure of X-rays, but it provides very accurate bond length information. So then we ask, were those bond lengths that were now measured by XAFS consistent with the structure that was in the crystal structure? And the answer is no, it doesn't match. So this is just a comparison, but you can see that it doesn't overlay very closely. It doesn't match the data in any of the states that are produced during this Koch cycle. And so we concluded that the crystal structure was not the native structure. What we then did was put in the correct oxidation states. Remember, I told you in the dark stable state, which is this S1 state, there should be 2 manganese 3 ions and 2 manganese 4 ions. So we put those oxidation states in and did a QMMM calculation and refined the structure and comparing it to XAFS data. And you can see the overlay is almost perfect now. And what you see comparing the crystal structure to the calculated structure is that the bond lengths all get shorter. Things sort of compress. And now the colored picture is the calculated structure, and you can see these atoms have moved. And in fact, now the calculated structure, the bond lengths all agree with what you'd expect from inorganic coordination complexes. And so we think we have a pretty good model now of the structure. But it's not coming only from the crystal structure. Of course, the crystal structure was essential as a starting point to be able to have the protein part of it modeled, and also as a starting point to position the atoms. But we really need to refine it by comparing to spectroscopic data. So the point I want to make is that don't believe necessarily the crystal structures, think about them critically, and in order to really get a full picture of this, you need to combine experimental data and spectroscopic data in particular with the crystallographic data. Okay, so let me then shift to how does this last step happen? So we're going to go around this cycle and look at this very last step. But this is where we get on very thin ice, because these initial intermediates, S0, S1, S2, S3, I told you, are produced and they last a long time. So they're very stable. We can make them. We can study them spectroscopically. But this intermediate, this is called S4, decays faster than it's formed. And what does that mean? It means it's not a kinetic intermediate. If it decays faster than it forms, it never builds up in concentration. And so there are no experimental results at all on this intermediate. And so how do you study it? Well, there have been computational models or just intuitive models that have been developed for how this reaction occurs, but we don't have any direct experimental data. So first what I'd like to do is tell you some of the models for how this reaction might be occurring, and then some new data that we've just obtained, it's not published yet, on looking at isotope effect measurements to probe the transition state for this reaction, which is, we think, maybe providing some new information about how that reaction is occurring. So here's some proposals. So this is now looking at the step where we make an oxygen-oxygen bond. You need to make two water molecules react with each other to form a bond. That's very hard to make that reaction start. And one idea that was published by us and also Vince Pecoraro in the mid-90s, well before the crystal structure was available, is based on some sort of, I guess I would call it sort of well-established chemistry, where one kind of reaction mechanism is a nucleophilic attack on an electrophilic center. Those of you who take in organic chemistry, you'll know nucleophilic substitution and all that. These are well-known kinds of reactions in organic chemistry and also inorganic chemistry. So the idea here is that we have an oxygen that's bound as a terminal ligand to manganese, where this very high oxidation state of manganese now sucks electrons out of that oxygen and it becomes electron deficient or electrophilic. And another water molecule is bound to calcium. Calcium, being a Lewis acid, will pull electron density away, make this water easier to deprotonate to generate hydroxide, and hydroxide's now a nucleophile. So you can generate a better nucleophile on one end, a good electrophile on the other, and we have a nucleophilic attack of an electrophilic oxygen. So that was a proposal that both we and Pecораro's group made. And in fact, we both proposed this well before the structure. When the structure came out, we were really pleased to see you've got exactly the alignment of waters bound to calcium and manganese, exactly what we would have had predicted. So this mechanism looks good on that front. But there's been also computational modeling, particularly from Per Sigban's group in Stockholm University, that suggested an oxo-coupling mechanism. And I don't favor this because from inorganic model compounds, these kinds of oxide bridge groups don't have any reactivity. So that doesn't look good from a reactivity or inorganic chemistry point of view. But it has been modeled from computational methods to be a lower energy route for the reaction. So this is the question: are these two mechanisms, one of them right, or maybe another one? How do you experimentally address that? And so this is where isotope effects may come in. So isotope effects are measuring, a kinetic isotope effect is looking at the relative rate that a heavy isotope reacts relative to a lighter isotope. So the idea is you turn your system over, produce the product lots and lots of times, and if you have a normal isotope effect, the lighter isotope goes a little bit faster then, and you get a little bit more of the product with the light isotope and a little bit less of the product with a heavy isotope. So this is a kinetic isotope effect. The reason that there's less of the heavy isotope is that there's a little bit bigger barrier for the reaction to occur with a heavier isotope. And that means we're probing the reaction barrier. That's the transition state for the reaction. And that's what we hope to be able to look at and learn more about how this reaction is happening. So this is not, we're not the first to look at this. The geochemists actually have been very interested in the fractionation of oxygen isotopes in photosynthesis for decades, because geochemists use isotopes to determine everything about the Earth's history. So they want to know what the isotopic composition of the atmosphere is and how it incorporates into rocks and sedimentary rocks and so on. And so they had measured this reaction, and this best result is from Joe Barry's group at Stanford University, and he concluded that there was no fractionation of oxygen isotopes in this reaction. And that seemed really strange to me. If you've got a system that's oxidizing water and you've got bond breaking and oxygen bond breaking, and you'd expect it to have an isotope effect. So we wanted to reproduce this, and so we did this. And we also, these earlier studies were done mostly on whole cells and some on membranes which were not completely purified. So there was a possibility of interfering reactions that could contribute. So we wanted to look at purified PS2, and using a technique of what's called isotope ratio mass spectrometry, which allows us to measure extremely accurately the relative amounts of heavy isotopes versus light isotopes in the product. And we did the reaction and measured it, got 1. Same result. The geochemists didn't screw it up. But then we started scratching our heads and thinking, okay, there wasn't any experimental reason why this number was 1, and so that wasn't an experimental error. But why is it 1? And then we started thinking about the overall turnover of photosystem 2. So photosystem 2 does water oxidation, but it also transfers electrons to quinones. And these quinones are organic molecules, and they diffuse in and out of the protein. And it turns out diffusion is a very slow process. So the diffusional process of quinones diffusing in and out is in fact the slow step in this reaction. And then it made sense. Obviously, quinone diffusion shouldn't have any oxygen isotope sensitivity, so that made good sense. So then we said, okay, we need to change the rate-limiting reaction. We need to speed up the quinone exchange or slow down the water oxidation reaction to make this now rate-limiting. So we did both. We can speed up the reaction here by using high concentrations of artificial quinones so that diffusion will go faster. This is a second-order reaction. And so remember, second-order rates depend on the concentration, so that should go faster. And it does. But we can also slow this down by using thermodynamics. So water oxidation is pH dependent. It gets harder to oxidize water as you go to more acidic pH. And that's reflected in the activity of the photosystem too. If you look at the oxygen evolving activity as a function of pH, it shows this sort of bell-shaped curve. It maximizes that around pH 6, and if you go below pH 6, the rate of the reaction drops off. And that's because it gets harder to oxidize water. So what we did was use low pH down here around pH 5, as well as high concentrations of quinones, and voila, isotope effect is manifested. And it seems to saturate out at about 1.022 at about pH 5. If you go lower, it doesn't change. And so we conclude this is now the isotope effect when we've made the water oxidation reaction fully rate limiting. So now we've got a new piece of experimental data. What does it mean about the mechanism? That's the big question. And so towards answering that, we're working with the Batista group to try to develop a computational model of the reaction pathway. And you can calculate an isotope effect from that. But we're not done with that yet. So that's work in progress. And then the next best thing is, let's look in the literature on values, isotope effect values for various possible reactions. So this is looking at now oxygen isotope effects here for ligand exchange reactions. And you can see ligand exchange reactions have very low isotope effects, not consistent. The number we measure is about 1.022, this line across here. So I think we can rule out that ligand exchange is the rate limiting step in this reaction. That wasn't expected anyway. We can look at electron transfer reactions, and they could have isotope effects that are in the range we measure. So that's one possibility, especially electron transfers that are coupled to protons, the so-called proton-coupled electron transfer. And that's because you have to break an OH bond, and that changes the bonding of oxygen, and what is important for isotope effects is the bonding, change in bonding of oxygen. So that could be one explanation. We see the oxo-oxo coupling. That was one of the two hypothesized mechanisms. Shows a much bigger isotope effect than what we measure. And so if that's rate limiting, which has been hypothesized by some groups, it seems like our experimental data would rule that out. And then finally, the nucleophilic attack mechanism that we favor also fits with the mechanism. So those are, right now we're sort of at this point where it could be either a water nucleophilic attack mechanism or possibly a prior step involving electron transfer. So those are things we're we're still working on, and so stay tuned. The picture isn't completely clear yet, but I think we're opening up some new approaches to probe this system. So that's the biology part. So those of you who are biologists now can go to sleep, and I'm going to talk about the artificial systems. But so as we think about developing artificial systems, we can ask, what lessons have we learned from nature? And I listed a bunch, some of which I won't go through because I didn't really talk about it. But two things that are important in thinking about how to design an artificial catalyst are that we think we should bind one water as a terminal ligand to a high valent metal ion to activate water as an electrophile. So that's something we can think about as a design principle for an inorganic catalyst. And secondly, we should react that electrophilic water with a nucleophilic water, and use maybe a Lewis acid is necessary to activate a water. So those are two things we've been thinking about in terms of designing catalysts. Okay, so let's shift gears now to our inorganic chemistry, and this is work that's being done at a new campus. Yale purchased the research and development site from Bayer Healthcare in 2007. We now call this our West Campus. It's about 8 miles from the main campus. So New Haven is back here, and that's where the main campus of Yale is. That's where our chemistry department is. But we've been setting up a new energy sciences institute, and our group has been developing now, doing research on developing artificial processes for solar energy conversion in the Energy Sciences Institute. And this is work that's being done in a collaboration with some of my colleagues. I mentioned Victor Batista already. He's a computational chemist. Bob Crabtree, an inorganic chemist, and also Charlie Schmuttenmeyer. I won't talk about his work. Here's Charlie here. He's a laser spectroscopist and helps us measure the photochemistry. And we teamed up about 10 years ago to try to work together to try to develop processes for solar fuel production. And we've been doing a lot of materials chemistry, interfacial chemistry. I'm going to focus today on catalysts, the water oxidation catalysts, in our efforts to try to design new catalysts that could do this reaction. And this overall process I'll call bio-inspired artificial photosynthesis, taking into account of the fact that we're using some of the principles that we think are being used in nature for natural photosynthesis, that is charge separation, water oxidation catalysis, and fuel forming reactions, to try to design systems that do analogous reactions, but not with proteins, but with more stable materials like metal oxide materials. And like, again, I'm not going to say anything about this, but I'm going to focus on this part here, the water oxidation catalyst itself, which is really a key problem. That's one of the more challenging chemical reactions you can imagine, and it still remains a bottleneck for developing good, efficient systems for doing solar energy conversion. Okay, so that's the idea. And now, I'll give you a little bit of introduction to this. So what we're interested in doing is making molecular catalysts. These will be inorganic coordination complexes that can carry out the water oxidation catalysis. And the very first one of these that was reported is this ruthenium complex. It's 2 ruthenium ions bridged by an oxygen and coordinated with bipyridine ligands. This is the so-called blue dimer that was reported by Tom Meyer's group at North Carolina in the 1980s. And for many years, this was the only example of a molecular water oxidation catalyst. In 1999, Bob Crabtree and I reported this manganese analog. So it has 2 manganese, 2 oxide bridges, a terpyridine ligand, and then terminal waters. Now, one of the things in common here is that we've got high valent, potentially, when we oxidize it, high valent manganese or high valent ruthenium and terminal water here, terminal water here. So those, one of the same kind of structural elements that we have in the photosystem 2 active site. So in 1999, when we reported this, these were in fact only 2 examples of molecular water oxidation catalysts that were known. And so that was kind of early days, but this has really exploded in the last 10 years. And there are literally hundreds, maybe thousands of papers now on water oxidation catalysts. I think almost every issue of the Journal American Chemical Society that you read now will have somebody's reporting some new water oxidation catalyst. The field has really exploded, and there's very many examples with almost every metal ion you can think of, but ruthenium is one of the more widely used one, but also cobalt catalyst, this polyoxometallate complex from Craig Hill's group, and cobalt oxide materials. Dan Nocera's group has pushed this idea of using cobalt oxides. So there's a lot of examples now. But one of the ones that caught Bob Crabtree's attention was this result that was published in 2008 by Stefan Bernhard. So Stefan Bernhard published this paper, and it didn't get very much attention because this is a very unstable complex, this organometallic complex. It very rapidly breaks down into iridium oxide, which is a well-known catalyst, heterogeneous catalyst, meaning that it's an oxide material for water oxidation. And it was widely believed, and in fact is correct, that this is not the catalyst. In fact, iridium oxide is the catalyst. So people mostly ignored it. But Bob Crabtree got incredibly excited about this because Bob is the world's authority on the organometallic chemistry of iridium. And all of his work has focused in the past on reductive catalysis using iridium. And so if you study organometallic chemistry, one of the things first things you'll learn is about the Crabtree catalyst, which is an iridium organometallic complex. So when this came out, Bob was jumping up and down. He said, wow, iridium could be used for oxidation chemistry. So we started working on iridium as an oxidation catalyst. And it turns out we've been able to develop some very good catalysts using iridium, molecular catalysts using iridium. But before I go into that, I wanted to just contrast the kinds of catalysts that could be either molecular or homogeneous, or oxide materials or heterogeneous. So this is an ongoing debate in the field right now as to whether the best route would be to make molecules that can do the catalysis and thereby do water oxidation, or materials, heterogeneous materials. And so what are the pros and cons? So as one of the advantages of molecular systems is that you can rationally design that. You can synthesize ligands with electron donating groups, with darring groups, make them more electron rich with electron poor. And so you can thereby tune them quite easily. And they're also in solution, so they're relatively easy to characterize using the methods we know how to use as chemists, NMR, X-ray crystallography, and these kind of methods that are giving us very detailed structural information. And oftentimes, molecular catalysts have the highest efficiency. But the problem with molecular catalysts is that they're not very stable. And particularly for water oxidation, this is a really big challenge. And to give you some point of reference, the bond energy for a water OH bond is about 118 kcals per mole. Okay? That's stronger than any CH bond. So that means if you have an organic ligand that has a CH bond, it's going to be easier to oxidize the ligand than it is to oxidize water. And every molecular homogeneous catalyst is going to have CH bonds, or most of them will have CH bonds. And so this is a big challenge. And as a result, molecular catalysts, if you're just carrying out the reaction in solution, will diffuse, and one catalyst will chew up the other. And they typically last for only minutes or less, and with turnover numbers that are typically in the tens to hundreds. Some are better, but none of them are stable. And so this has led many people to believe that it will never be possible to have molecular catalysts that really work for a long time. And so they want to use oxide materials. They have the advantage they're much more stable. But the problem is they're oftentimes not very good catalysts. They have low efficiency. And you can't synthesize materials. You can't make a coordination structure of a material. So this has been a challenge. And in my view, we need to overcome the problems of heterogeneous catalysts. And I'd like to show you how we've done that. We have used a high efficiency catalyst and have overcome the instability by putting these catalysts, anchoring these catalysts on a surface. So they're anchored on a surface. Now they can't diffuse and eat each other up. And that has allowed us to get molecular catalysts that are very highly efficient but last for millions of turnovers and months or longer of catalysis. So we think this may be the way to go in the future, is to optimize the catalytic efficiency by making a molecule and then anchoring it on the surface so it doesn't have the instability problem. So I'll tell you that story. And so this is where it starts. So this is our best pre-catalyst. This is one we started out with. It has a ligand, this pyridine alkoxide ligand, which is a chelating group, which holds on to the metal center. And we initially designed this with this ligand called CP star, which is this pentamethyl cyclopentadienyl ligand well-known ligand in organometallic chemistry, but it was a bad choice here because this is not very stable. It's very rapidly oxidized under the conditions we use for water oxidation. And this is a pre-catalyst because it's not the structure of what the catalytically active species is. We can make that catalytically active species by taking this pre-catalyst and either by oxidizing it electrochemically or using a chemical oxidant, we can generate an active molecular catalyst. This is a hypothesized structure. I'll talk about that more on the next slide. But we call this our blue solution. So it's a solution, but it's not a single molecule. It's a family of molecules that are all iridium dimeric structures like this with a bridging oxygen, but there's lots of different arrangements of ligands around them. This blue solution, however, is very active for water oxidation. It's very stable. And we can make this blue solution, leave it on our bench well, we've left it there for months at room temperature. It doesn't decompose. And so that's great. The problem is that we've tried to purify the active catalyst out of this solution, and we've not yet been successful to do that. So that's been a challenge. We have, however, used spectroscopy to characterize it. So this is using XFs and also a technique called high-energy X-ray scattering, which measure the distances between the metals and the ligands and metal-metal distances. And combining that with theoretical models, so the Batista group and X-ray analysis gives us a solution structure, even though we don't have a crystal structure of this. We, I think, have a pretty good determination of its structure. And this was published earlier this year in JACS. This is what we think the core looks like. It's got an iridium oxide bridge to another iridium and terminal water ligands. Again, terminal water ligands are the key thing for having a good catalyst for water oxidation. Now, that's a structure that has been derived from spectroscopy. We're also working to try to make these kinds of molecules directly from de novo synthesis, but not by purifying something out of this messy blue solution, which is probably a mixture of 100 different things or more. Instead, we're taking these iridium mononuclear starting materials, and I'll focus on this one today, which has 2 of these chelating ligands and 2 chlorides, and we're treating this this mononuclear precursor with silver oxide. So silver, you may know, silver chloride is very insoluble. So silver pulls off these chlorides. It has an oxide, which can then replace with an oxide. So we can make these dimeric structures that are like the ones we've hypothesized in the blue solution. And so we can then synthesize them, crystallize them. Here's one example. And so this is a solution. We can run it out on a thin layer chromatography, get all these great colors. And so some of them are dimers. We see these various blue bands. Here's a crystal structure of one of them. We have a whole family of them now crystallized and characterized. And this now gives us a way of doing very systematic structure-function comparison. So we can synthesize a well-defined molecule, test its catalytic activity, test another one. I can tell you that's early days because we've synthesized about 8 or 10 of these dimers, and none of them are anywhere close to as active as the blue solution. So something in the blue solution is a lot better than the ones we've made so far. So we've got a ways to go to actually find one that's going to really do. But obviously there's a whole range of catalytic efficiencies, and some of them are lousy, and some of them are okay, and some of them are going to be really good. And we're expecting that something in this blue solution is just, knocking our socks off as far as catalytic activity. We're really interested in trying to figure out what that is, and so that's ongoing. Okay, now I told you I was going to put this on a surface and show that it's now not going to decompose or be unstable. So this is what we're going to do. We're going to take this blue solution species and anchor it onto a metal oxide, and this is what I'll call a heterogenized homogeneous catalyst. So it's a molecular catalyst, but it's put on a surface, so now it's become heterogeneous. And so this is this blue solution species, at least the model of it from the spectroscopic model, and we're going to stick it on a surface. It turns out this is easy to do because it sticks on the surface all by itself. We don't have to do any kind of coordination chemistry or anything. You just soak it, a metal oxide, in the blue solution, and film sticks. So here's titanium oxide, tin oxide, tungsten oxide, iron oxide. All of them, it binds spontaneously. So one thing we can stick it on is an electrode. So this is a material that's conducting. It's called indium-doped tin oxide, which is a conducting oxide. And we can make it nanoparticulate, so we got a very high surface area. And that is now serving as our working electrode. We can stick our blue solution species on there, and we can carry out electrochemistry. So this is a technique called cyclic voltammetry. As we ramp up the voltage, we can see current passing as we go through, in this case, a iridium 3,4 oxidation. And then we can see this onset of current, which is coming from catalytic. This is called a catalytic wave in electrochemistry. And the amazing thing is that this is happening right at the thermodynamic potential, which means there is no overvoltage for this. And typically when you have a catalyst, you have to, for electrocatalyst, you have to apply a voltage that's significantly higher than the thermodynamic voltage to make the reaction happen. This is called an overvoltage. And for water oxidation, the typical value is about 400 millivolts. So 400 millivolts above the thermodynamic potential is about 1.1, would be way over here. But you can see that the reaction is happening right at the thermodynamic potential within 15 millivolts. That's remarkable, not observed in any other water oxidation catalyst. And it's stable. So this is now just looking at oxygen being produced as a function. We're just keeping a constant current flow. And over time, this is over 11 hours, and you can see it's constant. Staff Sheehan, who's the graduate student who's working on this, has actually carried out this reaction for months, and it continues for months with no loss of activity. And the calculated number of turnovers with about 8 per second is in the millions. And we've also analyzed, in this case after about 100,000 turnovers, whether or not the structure has been changed. And so this is using a spectroscopic technique called X-ray photoelectron spectroscopy, which allows us to probe iridium structure, the nitrogen ligand structure, and the carbon in the ligand structure. And all of these are exactly the same as we started. There's no measurable degradation of the ligand. It's still bound, and so this is really exciting for us, that this is an example of a molecular water oxidation catalyst that's now, as far as we can tell, indefinitely stable on a surface. That's pretty exciting. Staff Sheehan was the graduate student who did this. He graduated in May. He's a great scientist, but he's a better businessman, and he decided to take this and start. So he has a startup company that from this result is called Catalytic Innovation. And he's actually making money after just a year. But it's not making money by doing water oxidation. Nobody cares about water oxidation. We've got plenty of oxygen around. We don't need to make it from water oxidation. But it turns out that this blue solution, when you stick it on surfaces, also prevents corrosion of the surface. So he's making big bucks preventing corrosion in pipes. And so he's a businessman now. So that's what Staff is doing, and he's got his company. Okay, so let me just end by coming back to manganese. So I'll just say a little bit. This is very early preliminary results on this. But I told you we had reported a manganese catalyst in Science in 1999. That was one of the 2 early molecular water oxidation catalysts. And then we shifted over to iridium. So why weren't we doing manganese? Well, manganese, it turns out, is really kind of a pain to try to make work because it's very unstable. So this manganese terpene complex, although it's the fastest turnover manganese catalyst known, we reported it in 1999, and no one, including us, has made one better. This is what Bob Crabtree likes to call the principle of initial optimization. So we made it the first one we made, it's the best one. But we're trying to do better. And one of the reasons this one doesn't work very well is that in the catalytic cycle, this is a chemical oxidant, we make this high valent manganese oxide species. It reacts with water to make oxygen, but then it makes this very low oxidation state manganese species with manganese 2. And manganese 2 is extremely labile, which means the ligands come on and off really fast. And so what happens in this catalytic cycle is the complex falls apart, and it has to be reassembled in order to complete more than one turnover, and it just doesn't last very long. So how could we do better? One idea is we could use a ligand that stabilizes higher oxidation states so we don't make this low oxidation state manganese. And that's what we think this pyridine alkoxide ligand will do, except we've made a different analog. So this is the ligand we've recently synthesized. It's a bipyridine alkoxide, so it's analogous to the one I just showed you, except it's got this extra pyridine group. and it's forming a tridentate ligand that allows us to make a manganese dimer very similar to the manganese terpene complex. If I go back, here's this manganese terpene complex. It has this tridentate terpyridine, which 3 pyridine groups. Now with this new one, we have 2 pyridine groups, but now an anionic ligand should help stabilize a higher oxidation state. In fact, it does. And we've synthesized this dimeric species here, which is containing 2 of these new pyridine alkoxide ligands, a dimanganese oxide structure, very analogous to this manganese terpene complex we had reported earlier, has terminal water ligands. And so that looked good, and what we're finding with this complex is that it produces oxygen over a very long time. This is after a week of turnover, and we can see we still are making oxygen at a reasonable rate. This is by far and away the most stable manganese-based water oxidation catalyst that's been known. The only problem is it's a really crappy catalyst. If you look at the turnover numbers, we went for a whole week, but we only still made a couple hundred turnovers. So we need to work on making this a faster catalyst, but it's very encouraging to us that it lasts this long. If we took the manganese terpene system that I showed you, the rate would just go through the roof, but then it's done after about 10 minutes, and this one lasts for a week. So this is encouraging, but it's still not ready for prime time yet. Okay, so that's where I'd like to end. I guess I went a couple minutes over. I want to just finish with saying where we're going in the future. We're interested in now putting these catalysts onto a surface for photochemical initiation and a complete photoelectrochemical cell for water splitting, so we can make oxygen and hydrogen, for example. And that's the solar fuel problem. We're trying to now incorporate some of these catalysts that we've been working on. So let me end with a little excerpt. So my colleague Bob Crabtree not only is a great organometallic chemist, but he's a book collector as well. So one of the books he collected is this book by Jules Verne called The Mysterious Island, which was written in 1870, over 100 years ago. And these guys are, they land on this island, and this is a little excerpt. He said, "And what will we use instead of coal?" "Water," replied Cyrus Smith. "Water?" cried Pencroff. "Yes, but water split into its elements, hydrogen and oxygen," responded Smith, "and no doubt split by electricity, which will have become a powerful and versatile force. For hydrogen and oxygen, its constituents will provide an infinite source of heat and light of an intensity unknown for coal. Water is the coal of the future." So I really believe that, and that's what we're working on, to try to turn water into a fuel source, and that's where we are. And so let me just end by thanking the people, and knowledge, everyone who did the students who did most of the work I talked about today were Julie Thompson and Sahar Khan and a postdoc, David Vineyard, and of course my colleagues Victor Batista, Bob Crabtree, and Charlie Schmittenmeyer, and all the others in our group at Yale. And thank them, and thank you for your attention. I'd be happy to answer any questions.
Student questions first? Even biology questions. Or physics. Fine, then how about faculty questions?
How long does it take you to come up with the models using a computer? And
So-
how much computer power do you-
That was my question.
Yeah. This is Victor's area, but so the DFT calculations that we're doing require pretty substantial amount of computational time. He has access to supercomputer time, and also we have quite a good high-performance computing center at Yale. But one round of calculations takes about 2 weeks, and to optimize it takes longer. So it's a slow process, and it's not the kind of thing you can do on your laptop in a few minutes. And the DFT calculation, for example, has all 5 of the metal ions, 4 manganese and a calcium, all of the amino acid ligands, plus some of the second shell protein residues and water molecules as well. So there's quite a few atoms. That's a big DFT calculation, and it takes a while. And so to get it converged, it takes us weeks, to converge one of these, or longer. And the problem with DFT, of course, is you have to come up with an initial guess, and that's really where I come in. Because if you just blindly pick stuff and do calculation, you'll be calculating forever, and you may never get anything that's useful. So you really have to rely on some insight into what is the right kind of calculation to do, and that's where I try to bring in my insight from the experimental work that we do. what is a structure, we have to identify things like where are the protons, we can't see those in the crystal structure, and what structural changes might occur as you go from one oxidation state to the next. Those are things that I think you can provide some insight from your chemical insight, and that makes it go a lot faster. So I think that has helped, but ultimately it's a slow process to do that. Yeah.
So for your artificial catalysts, you're using electricity now to provide the energy. Do you envision a big problem shifting from that to light?
Well, you actually touched on a really big topic of discussion, and so this is the question of, do we want to do a solar fuel process in a direct fashion, which is, do we want to have light directly coupled to the catalyst to do the water splitting chemistry, for example? That's how nature does it. Or do we want to have an indirect process where we just have a big solar farm of photovoltaics to make electricity, and then we have a centralized electrolysis facility to do the catalytic reaction? And there are pros and cons. So in the direct process, there have been calculations on efficiency, and it's the more efficient way to go, because every time you make more interfaces, you have more losses. And so that's been demonstrated, at least theoretically, that if you have a direct system, it would work better. But on the other hand, it makes it very hard if you're going to, let's make hydrogen, for example. To collect hydrogen over, a 1,000-acre farm, it's not so trivial, right? And so how do you flush all that hydrogen into some place where you're going to use it? As opposed to an indirect system where you just use wires to conduct electricity, and now you make a lot of hydrogen. That could be more practical. On the other hand, if you do a direct process, the rate that sunlight hits a given absorber is relatively low frequency. The photon flux, if you have just, say, one chlorophyll, that chlorophyll absorbs about one photon in a few seconds, only every few seconds. You don't need a very good catalyst to keep up with something that's only going one every few seconds. But if you've got a 1,000-acre photovoltaic farm feeding electricity into a central catalytic center, you've got to have a hell of a good catalyst to keep up with that. And so that puts really high demands on the catalyst. And so the people who make lousy catalysts tend to think, well, probably the direct system is better. So we've been trying to make really good catalysts, and maybe in the future, my own thought is probably the indirect route will be more practical in the long run. But we are trying to develop direct methods too. So obviously that's of interest, and we're going to work on that. But it may turn out that the catalyst, like the one that we've used already, which is electrocatalytic, might be already what we need, and we just take photovoltaics and plug them into this, and we're good. Yeah, so that's, I think, these are questions for the future, but we don't have anything that's really practical yet, so I think there's a lot of areas that need to be explored before we know the answer.
All right, let's thank Dr. Brudvig one more time.
Thank you.
For more information, please visit tlu.edu.